Hello, welcome everyone to the Arthritis National Research Foundation's Researcher Spotlight Series. I'm Laura Douglas, Grant Program Manager for ANRF, and on behalf of our Board of Directors, Scientific Advisory Board, and staff, I would like to welcome you on the Spotlight of Osteoarthritis. We are excited to have so many of you joining us on this call from around the world. We would also like to recognize and thank all of our corporate sponsors. Thank you, Amgen, for being a gold level supporter of the series, and thank you to AbbVie, AstraZeneca, and AOPI for your continued partnership and dedication. It is because of our partnerships that we are able to continue to pave the way for new innovations and learnings from our researchers. Thank you. Before we get started, there are a couple logistical pieces of information we want to share. First, this webinar is being recorded, and we will have it available on our website and YouTube channel in the coming weeks. If you have any questions, please type them into the Q&A. If we aren't able to get to yours, please email info@curearthritis.org after the webinar with your question. I am now honored to turn over the mic to our patient moderator, Tracy Augustine, to tell us her story of living with osteoarthritis.
Hi, everybody. My name is Tracy Augustine, and I will be your patient moderator for the day. Thank you for coming to our Zoom today. We're excited to meet and interact with all of you. I'm a patient advocate and volunteer with the Arthritis National Research Foundation. Fundraising for arthritis is my passion. I organize 2 yearly fundraisers, which allows me to give back to our community, and that means so much to me. I've traveled to Washington, D.C. and my state capital multiple times to advocate for arthritis legislation. A little bit about me. I was diagnosed with early onset osteoarthritis in both of my knees at the age of 31. In addition, I'm also diagnosed with rheumatoid arthritis and fibromyalgia and a bunch of other medical things. I have a family history of early onset osteoarthritis, but I didn't know that until after I was diagnosed. I actually started having knee pain when I was 25. I was diagnosed at 31, but at 25, I dismissed the knee pain as just, oh, I'm sitting at a desk all day for work and my knees are going to hurt. Osteoarthritis was a big contributing factor for me that required me to have a knee replacement at 39 years old. For years, I was treated with gel injections and steroid injections, and they worked until they stopped working. And even with the injections when they were working, I never had full pain relief. I tried 2 rounds of physical therapy for both of my knees, but the pain relief was temporary. The pain in my right knee progressed over time, and it started to affect the quality of my life. My apartment that I live in has a flight of outdoor steps to get to my front door. And at the end of a long day, I could barely make it up the steps. And there were several times where I debated sleeping outside. It was time for a knee replacement surgery when I could only leave my house once a day. Mind you, this is when I'm 38, 39 years old. I could only leave my house once a day because walking up and down those steps to get to my door was so painful. I struggled to find a doctor to operate on my knee due to my underlying medical conditions. There were days where I thought I would have to move to another apartment on the first floor to accommodate my constant knee pain. And if 2 things don't go together, it's arthritis and moving. They really just don't go together. Eventually, I found an amazing surgeon who ordered x-rays, and the x-rays showed that I had tricompartmental arthritis in my right knee. I had it replaced in September of last year. And yes, I did celebrate this September with my 1-year anniversary with my new knee. I didn't realize how much pain I was in until I have the replacement, and I'm so grateful I was able to have it. I'm excited to volunteer with an organization that is funding important and necessary research for osteoarthritis. I hope in the years to come that this important research will lead to early detection and better treatment options for osteoarthritis. And while I don't have any formal education or credentials to add to this, for some reason, I thought it was important that in my spare time, I spend time with my cat, and my favorite food is melted cheese. And I would like to thank the Arthritis National Research Foundation for including me in today's panel. I would like to introduce our first guest for today. We have with us Dr. Michael Jurynec. and he is an assistant professor in the departments of orthopedics and human genetics at the University of Utah. His lab uses a combination of approaches, including human genetics, bioinformatics, and model organisms to discover the genes and pathways that contribute to osteoarthritis. We welcome Dr. Jurynec today. Thanks for joining us. How are you doing today?
I'm great. Thanks for the
Okay.
introduction, and thanks for that wonderful speech.
Oh.
I'd like to thank the ANRF for inviting me today, and also for providing support for my research. I think it's very, very important, the support they give to young investigators. So today, I'm going to tell you about how my lab uses humans and mice to identify new targets for OA therapeutics. So my lab, as Tracy said, I'm at the University of Utah Department of Orthopedics and Human Genetics, and my foray into osteoarthritis was very similar to Tracy's. I was diagnosed with early onset OA in my early 30s, and realized it ran in my family. So my grandma, my dad, my brother, myself all had the same phenotype. We all had arthritis in our foot, and being a geneticist, I was really curious as to why we all have this disease and why others develop this disease. And this really laid the stage for my interest in my research in trying to understand the biology and genetics of OA. And on the screen there, this is a radiograph x-ray of my foot, showing on the bottom there, the loss of joint space, typical OA type symptoms with the joint space reduction and presence of ectopic bone formation. So we know osteoarthritis is a very painful, debilitating disease, which involves this abnormal remodeling of your joint tissues. And this is the hip, knee, your hands, shoulders, elbows. And it is the major cause of disability among the aging population. It affects young people, it affects old people, it affects a lot of us. Over 240 million people worldwide and 32 million in the US alone. And despite a lot of interest in studying the disease and trying to understand why we get it, we have no drugs that inhibit the progression of the disease. And this is a major medical issue. So why don't we have drugs that stop progression of the disease? Well, OA is a complex disease with many missing parts. We know that there are many factors that can increase your risk for developing osteoarthritis, such as injury to your joints, congenital abnormalities with the way the joint develops, and also things like genetics, which is what I'm interested in. And so my lab takes an approach to try and understand what are the genes, which we all have in our cells, which make proteins, which allow our cells and tissues to function normally. How do changes in this DNA cause osteoarthritis? And I believe this is important from several standpoints. First, it'll help us understand why all these other risk factors, how all these other risk factors contribute to development of disease. It allows us to develop new targets so we can actually start to make drugs that inhibit these genes that are defective to hopefully stop the progression of the disease. And finally, it helps us understand the biology of the early phases of disease, which will help us identify those who haven't yet developed symptoms. And for example, we know that there are many cells that make up the healthy joint. So this is just a schematic of the knee joint here, showing this cartilage in purple, bone in blue. So we know what a healthy knee joint looks like. We know what end-stage osteoarthritis looks like. And you can see that you lose cartilage, you get ectopic bone formation, you get inflammation. But we really don't understand what are the primary or initiating factors that cause the disease. And so again, my interest is really trying to understand what are the genes, what are the genetics that are this question mark. And so the way we go about doing this is we want to study families with osteoarthritis. And we have this very powerful tool in Utah called the Utah Population Database. And it's really the only database of its kind in the US, and one of very limited resources in the world. And what it is we have over 11 million people who can be linked to basically these large families down below. And we can actually link these people back to medical records. So we can actually find individuals who have various forms of osteoarthritis, and then find all their other family members, which is very powerful to do genetic studies. And the population database has been used to find many disease genes. And so how do we use this population database? So we focus on several forms of OA, including OA of the foot, shoulder, and hand. We find individuals, we map them back to these, what we call pedigrees or family structures. And these little red dots are those people who have arthritis. And then what we do is when we contact these people, recruit them for our study and say, we'd like to study your DNA and understand why you get the disease. And so we've done that to date with over 150 families. And what this gives us is specific genes and pathways that are disrupted in each family. But then as we analyze more and more families, we find common themes, okay? And those common themes become important for identifying and testing new drugs. But first, we have to determine how those changes we find in families cause disease. And we can't do this in humans, so we use the mouse. Mice have joints that are almost identical to ours, and we have really cool tools in the mouse where we can actually put in the single mutation we find in our human families into the mouse and make the mice hopefully get arthritis. And this allows us to test drugs and understand the biology of the disease. So this is just one example of a gene that we identified in a family. We put this specific mutation into the mouse and then asked, does this cause accelerated OA? And again, this is just a schematic of the knee. This is an actual section of a mouse knee. And this purple area is the cartilage. In mice, just like us humans, develop arthritis as they age, and this is indicated by loss of this purple staining. And so this is a normal old mouse. When we put that disease gene into the mouse, we now see that they have very, very severe OA. Okay, and so this tells us that this disease gene is causative in our human families. And again, this allows us two things. It allows us to understand how this gene works, and it allows us, once we identify drugs, then we can test these drugs in the mice and hopefully translate that to humans. And so this is exactly what we're doing. So from our family studies, we have a number of high priority candidate genes and pathways that we're testing. And for example, one is to this RIPK2 gene. We have collaborated with a company who has inhibitors that block the function of this gene. And we're currently testing this in our mouse model to see if we can either prevent or slow the progression of OA in our animals. And another pathway that we've found disrupted in many other families is one that involves inflammation and oxidative damage. And so we are testing now a number of novel compounds to see if we can, again, inhibit this pathway and see if it prevents disease in our animal models. And again, hopefully once we prove that, then we can translate these studies and potentially move these into the clinic in the future. And so with that, I'd like to thank members of my lab who have done all the work, the funding, especially the ANRF, who has been very supportive of my work and allowed me to establish myself as an early stage investigator, and then a number of other collaborators who have been extremely valuable in helping collect families and do a lot of the work that we've done here. So thank you, and I'd be happy to take any questions.
Thank you so much, Dr. Jurynec. I did have a couple of questions, and these are some pre-gathered audience questions. Do you happen to know in your research, are there common co-diagnoses with osteoarthritis?
Yeah, certainly there are many factors that are often associated with OA. So for example, obesity is often linked to OA, injury. So if if you've torn your ACL in a ski accident, for example, that's you're more likely to develop OA. So yeah, so there are many risk factors that contribute to development of the disease. It's not fortunately, it's not it's not a binary switch. So having these risk factors only slightly increases your risk. It's not to say that if if you're obese, that you're definitely going to develop OA, for example. So these are mostly modifiable risk factors. So you can obviously change your diet. You can exercise, right? Those are certain things that can be changed to help prevent development of OA. Good question.
Okay. And are there any potential interactions between OA and other health issues? Have you noticed a pattern?
Yeah, again, certainly that there are many health issues that contribute to development of OA. In our studies, we primarily focus on families. And so we try and minimize those other risk factors. So we try and exclude people who have injured their joint, because we want to understand what are the genes that cause OA, not how injury causes OA. But certainly they are interrelated, and I think understanding the genetics will help us understand why people who injure their knee might be more susceptible. So it might be that the genetics affects other health conditions.
Okay.
So that's how we're approaching
I'm sorry. Are you currently looking to expand your study for more patients?
Oh, absolutely. Yeah. So we, if anyone's interested, if you have a family history, please reach out to us. My website, juryneclab.org, has my contact information or contact information for a clinical coordinator who we'd love to talk to you. And certainly we would be definitely interested in recruiting more people if people would like.
Okay. And just so everybody knows, Jurynec is spelled JURYNEC lab.com.
.org.
.org. I'm so sorry.
Yeah, no, that's great. You can Google
I knew it was a
too.
dot something. And then what do you think are the future trends for osteoarthritis research?
Yeah, it's a great question. I think you brought up some of those points in your introduction. I think one of the main things that I find very exciting about is there's two areas. One is identifying potential biomarkers. So these are things that we can measure, whether in your blood or your urine, that may say that, hey, you're probably going to develop OA. So I think that's a very exciting part of the OA field. And then also, which goes along with that, is identifying ways of diagnosing people before they have severe joint damage. So if you have, for example, if you have a family history, we potentially develop a panel that we could use to say, okay, well, your dad had OA, and so let's test you and see if you may develop OA. And so then we can intervene before you have to get to that, before you're at that stage where you develop pain and have to go see a provider and potentially get a joint replacement. We want to mitigate that. And so I think those two, early detection and biomarkers, I think are very interesting avenues of research that I think are ongoing, and I think will be very informative.
Okay. We have an audience question. Do you feel that OA and AS are interrelated?
Yeah, so OA being osteoarthritis, and AS, I believe, is referring to ankylosing spondylosis. Yes. Yeah, so we're finding, so with the rheumatic diseases, so the OA, AS, rheumatoid arthritis, they're generally thought of as really distinct diseases, but we're actually, in terms of the genes and pathways that we're identifying, we are finding some overlap with among the different diseases. So yes, I believe that at least genetically, there is going to be a small degree of overlap between the different diseases. In particular, those genes and pathways involved in inflammation.
Okay, perfect. Thank you for explaining all of this to us too, from the perspective of a research scientist. And if, let's say I'm having, it's pre-diagnosis of what's really firm, or I have osteoarthritis, what questions should I be asking my rheumatologist? So, for example, if I have a family history, what should I ask? And then if I'm having pain, what should I ask?
Um, that's a good question. I'm a scientist, so
Mhm,
in terms of, but I do have OA. So
mhm,
I think the family history is important, and I think asking your provider, what they would suggest in terms of lifestyle modification, because obviously, as you know, right, we don't have good drugs, right? You
mhm,
can take, pain relievers, but those only work so well, right?
mhm,
And so I think going, if you do have pain in your joints, I think seeing a provider sooner is better, and really asking about lifestyle modifications, what they would recommend
mhm,
to help mitigate some of those painful symptoms. I think those are getting there early, and then talking about those lifestyle interventions are probably two of the more important questions that I would ask as a patient myself.
mhm. Okay.
Yeah.
Those are good. And then I had a question of that I thought of last night. A friend of mine has really bad osteoarthritis in her knees, and she lives on a split level home. So when you walk in, she has the living room, upstairs are the bedroom, and then downstairs in the basement is where she lets the dogs out. Not the
Uh-huh.
song, but the actual dog. So, um, what could you think of any home modifications that you could in put in place? I know it depends on the body part. Um, that like you could recommend if maybe you have a family history and you want to mitigate some joint pain.
Yeah, I think certainly, preventing those sharp inclines and declines would likely help. So putting in some ramps, for example, I think would be
Mhm.
very helpful. Yeah, I myself, I have problems in my toes, so I wear, relatively stiff footwear that seems to help. But yeah, certainly those steep inclines, you want to try and reduce those as much as, or inclines and declines as much as possible.
Mhm. Okay. We have another audience question. Can a person who is adopted with no medical history provide with, so she was adopted, her medical history from either side of her family wasn't given, and now has 5 types of arthritis. Can she be considered for your study?
Certainly, if she'd like to reach out to us, we would love to talk with her. Yeah.
Okay. All right. Thank you so much. And do we have time for one more? Yes. So, how can I stay up to date with the latest research developments in your area? We gave the website, but can you think of other places or websites to go to where we could be informed about more OA research?
Yeah, certainly ANRF provides a lot of those links on their webpage. I think just simply using Google,
Mhm,
just Google osteoarthritis research. I think that will really keep you up to date. And I think we could provide, there are several other foundations which provide more patient-based uh, resources. And I wonder if we can potentially link that. I don't have those with me right now, but I wonder if
of
we can
course.
potentially link that. And I think
Mhm,
those would also be helpful to people. There are webinars that are given throughout the year, which I think are patient focused, which would be interesting.
All right. Well, we can wrap up. Thank you again so much for your time. We really appreciate it and your willingness to discuss this from a researcher's perspective and also the patient perspective. So, thank you so much for joining us. We really appreciate it. I would like to introduce our next researcher, it's Dr. Amanda Ali. She's been studying osteoarthritis for 15 years. She completed her PhD at the University of Toronto in 2014, followed by postdoctorate fellowships. In 2020, she became an assistant scientist at the Henry Ford Health in Detroit, Michigan, where she leads a laboratory using human tissues and mice models to better explore ways to diagnose and treat osteoarthritis. Everybody, please welcome Dr. Ali.
Thank you again. It's really a pleasure to be here. And today I'm going to talk to you about some of the research that's been supported by the Arthritis National Research Foundation, for which I'm super grateful. As an early career investigator, this type of funding really helps us get off the ground and get our research going. And so I have a complicated title on this slide, elucidating the role of microRNA 126.3p in osteoarthritis. But my goal today is to break this down for you and have you walk away with a little bit more information on some of this new research that's being conducted. So first about me, again, I'm Amanda, and I was actually born in Guyana, which is a small country in South America. But my family immigrated to Canada when I was just 2 years old. So for all intents and purposes, I'm a Canadian, and this is where I've done all my training, as Tracy just mentioned. But I'm currently working at the Henry Ford Health System in Detroit, Michigan. And this is where my research program is currently focused on osteoarthritis. A fun fact about me, I'm a first-generation scientist. What that means is that no one else in my family has pursued a career in science. In fact, no one else in my family has a PhD or even an undergraduate degree, so I'm also a first-generation academic. But this doesn't mean that my family hasn't had a huge influence on my career path, because the whole reason that I study osteoarthritis in the first place is because of my grandmother. So I'm showing you here a photo of my grandmother who is now passed away, and I miss her dearly every day. But she suffered from osteoarthritis for 4 decades, and watching her slowly decline over time really motivated my interest and passion for understanding osteoarthritis. And as Mick mentioned, there's no cure for this disease. There are no good treatments for this disease. So we really want to better understand what's going on here so that we can help the millions of patients who are afflicted by this particular condition. And so I'm going to tell you about the goals of my lab, and they can be grouped into 3 categories. First, we're interested in diagnosing osteoarthritis. In particular, we want to diagnose osteoarthritis in earlier stages when we can still prevent the disease from becoming worse. So we can have interventions that are preventative. Second, we want to understand osteoarthritis. So I'm showing you a very complex map of a bunch of different molecules that are all interacting, only to say that this is a very complex disease. And again, I'm echoing some of the comments that's already been made by my colleague, but it merits being repeated because it is really a difficult problem to solve. And we really need concerted efforts to understand all of the different players that are involved in osteoarthritis. And finally, with that understanding, our lab hopes to develop better treatments for osteoarthritis. Now, whether this could be something that we inject, as shown here on my slide, or other type of preventative interventions, as mentioned, exercise, we're hoping to look for better treatment strategies, again, to reduce the burden of this disease in the population. So I'm going to tell you a little bit more about each one of these stories today, beginning with diagnosing disease. So biomarkers have already been mentioned, but what really are biomarkers? These are markers that are found in the body. It can be imaging, but it can also be found in blood and urine and all other biofluids. And they can help us understand if a person has a disease or the severity of the disease a person might have. So I'm showing you here the Kelvin-Lorentz radiographic grading scale for osteoarthritis, and this is really widely used to diagnose disease at the current time. So what you can see is grade 0, individuals with grade 0 do not have osteoarthritis, so they would have what we would call a normal knee. And as you progress with each grade, you get to more and more severe osteoarthritis. So you can see by grade 4, this person has the joint space narrowing that Mick also mentioned as he showed us in his foot. You can see that it's here in the knee joint. And we also see some subchondral sclerosis, which just means the bone is becoming thicker. And you can appreciate these types of things on an X-ray in addition to bone formation and other types of features. But the Kelvin-Lorentz radiographic grading scale is limited in some aspects because we can only really reliably diagnose the disease once it's established, once it's already there. So the question becomes, how can we find osteoarthritis in earlier stages, as I mentioned, when we can still have opportunities to prevent it from progressing? So my lab is really interested in something called microRNAs, and we're interested in them in each of the 3 capacities I mentioned earlier, and so I'm hoping to walk you through that today. So we know that DNA encodes RNA, and RNA can become protein, and that's the essential foundation for everything in our body and how all biological systems work in our body. But the role of microRNAs are to block specific RNAs from becoming protein. And in doing so, they're actually very important in a lot of the biological systems in our body, including a contribution to osteoarthritis. So the approach that we take in our lab is to collect a patient cohort. So we currently have close to 500 different patients with varying degrees of knee osteoarthritis, as shown here. What we've done is we've collected blood samples as well as other tissues, but I won't be talking about that today. We have blood samples that we're able to isolate microRNAs and then subject those microRNAs to sequencing. Now, sequencing is this fancy technology that we use that's not so new anymore, but is really helpful for reading all of the microRNAs that are in a particular sample. So again, here in the blood, and it can tell us which microRNAs are associated with people who have osteoarthritis as compared to those who don't. So I'm going to summarize a whole bunch of work and very quickly just tell you one of the microRNAs we found is microRNA 126.3p. Now, this particular microRNA appears to be really promising for us, and I'm going to show you why. So we measured this microRNA in the patient population that I just mentioned. So although we've gathered up to 500 samples, we started by looking at around 100 patients, as you can see in this graph. Along the bottom of the graph, you can see Kellgren-Lawrence grades, as I just mentioned. And along the side of the graph, you can see what we're measuring is microRNA 126.3p. And I've added this little vial of blood to remind you that this is all being measured in the blood. So these are really easy samples to access from patients. So what you can appreciate, hopefully, is that at Kellgren-Lawrence grade 0 and grade 1, which are pretty early stages of osteoarthritis, we do not see levels of this microRNA in the blood. But at Kellgren-Lawrence grades 2, 3, and 4, you can see that there is an increase in the level of this microRNA, suggesting that this microRNA could be used as a potential marker to detect Kellgren-Lawrence grade 2 and onwards, and potentially has a role as a biomarker for osteoarthritis. So I hope I've convinced you that we can use microRNA 126 potentially as a biomarker. But of course, what is that microRNA doing? We want to understand now what role it might be playing in disease, given what we know about how microRNAs work. As I mentioned, we know that microRNAs are able to block RNA from becoming protein and therefore play important roles in biological processes. So we looked at previous studies that had investigated microRNA 126. And although there are no previous studies looking at this specifically in osteoarthritis, there are previous studies that have looked at it in vasculature. So vascular systems means things like the heart. And what those studies have found is that microRNA 126 can block a specific gene called SPRED1. And when it blocks that gene, what happens is that there's an increase in angiogenesis or blood vessel formation. So I'm trying to show that here visually in the knee, because what we're hypothesizing is that potentially when microRNA 126 becomes elevated in Kellgren-Lawrence grades 2 and onwards of knee osteoarthritis, the effect of that is potentially to increase angiogenesis. And we're still investigating whether the overall effect of this is good or bad for osteoarthritis. That we're not sure of yet. But for now, what we've determined is at least microRNA 126.3p is indeed regulating SPRED1 in knee osteoarthritis. So now we're wanting to understand if we can somehow target microRNA 126.3p in order to understand if it can be used as a treatment. So one of the really neat things about microRNAs is that they can be modulated with small molecules. That means we can block them or we can increase them with small molecules. So what I'm showing you here is that a microRNA typically binds to the mRNA target. But when we have these small molecules, given this fancy name here called antisense oligonucleotide, it, as you can see, binds to the microRNA and prevents it from attaching to its RNA target. So effectively what you're doing is blocking the function of the microRNA. And while I haven't shown it here, you can also add more microRNA to a system if what you're wanting to do is increase its activity. So what we're looking at right now is a mouse model of knee osteoarthritis where we are injecting either the microRNA or a blocker against the microRNA. And we're looking to see if this has an effect on knee osteoarthritis outcomes. And this is called a preclinical trial. So what we're doing is looking at the effects on knee osteoarthritis, with the hopes that we're going to see some kind of benefit if we're either increasing or decreasing the microRNA. So what I hope I've convinced you of in this very short time is that microRNAs really have immense potential to help with diagnosing, understanding, and treating osteoarthritis. And with the support from the Arthritis National Research Foundation, our lab has been able to identify microRNA 126.3p as a really promising candidate that we're further investigating. Now, I have to give recognition and acknowledgement to all of the people who contribute to this work. As shown here, this is my current lab group at our building in Detroit, Michigan. And if you have any questions, I'm happy to take them now. But if you think of a burning question later, I've also left my email address on this slide. And I'm also on Twitter, so feel free to follow me there. Thank you so much. And again, I really appreciate the opportunity to share my work with you today.
Thank you so much, Dr. Ali. There is one question in the chat. The mRNA strand of 126, could that be used to differentiate between different grades of osteoarthritis?
That's a fantastic question. And currently, it appears that the microRNA becomes elevated at Calvert-Lawrence grades 2, 3, and 4 at about the same level. So I don't think it can be used to differentiate between those 3 grades, but I do think it can be used to differentiate earlier stages of disease from later stages of disease. So we do not see it elevated at Calvert-Lawrence grade 0 and 1. But we do see this sharp increase at Calvert-Lawrence grade 2. So if you want to think about it as buckets, right, we can have a bucket that we call Calvert-Lawrence grade 0 and 1, and then a second bucket where we call 2, 3, and 4. So it can be used to distinguish those 2 buckets, but I wouldn't say that it could be used to differentiate the individual grades. There are other microRNAs that we're looking into that might have more specificity to the specific grades, but I wouldn't say that's the case for 126 in particular. Thank you. That's a great question.
And we have another audience question. Are there also other biomarkers that you have identified that are worth studying?
Well, thank you for the opportunity to talk about my other work. I could go on and on, of course. So one of the other really interesting studies that I'm very excited about is looking at fast progressing osteoarthritis. So we have found some microRNAs that are uniquely associated with individuals who become diagnosed with osteoarthritis and then very quickly end up needing a joint replacement. So this means they go from Calvert-Lawrence grades 1 or 2 to 3 or 4 in a period as quickly as 4 or 5 years. And so we're wanting to understand, why does that happen? And why are there others who can have Calvert-Lawrence grade 1 or 2 for 20 years? And they progress much slower. So that is another set of microRNAs that I'm really interested in. And some of that work has been published. And so if you're interested in reading about that, of course, it's going to be a little bit more difficult to wade through all the details, but that study is published, and that is something that we're interested in pursuing as well. So thank you for that question.
We have another audience question. Do you think different tissues, for example, blood or urine, will be more informative with relation to KL grade?
That's also a fantastic question. And so microRNAs are found in all tissues and all biofluids. So arguably, we could look at microRNA levels in any given tissue or biofluid. So the reason, first of all, we choose blood is because it's really easy to access. But of course, so is urine. Urine is also easy to access. Synovial fluid is a little bit more difficult. And so that kind of provides a little bit of rationale as to why we're using blood. But I do think that different profiles exist in the different biofluids. Here's what we know about microRNA 126.3p. In the patient population that we've collected, not only do we have plasma, as mentioned, blood plasma, but we also have synovial fluid and several tissues. So we've looked at the synovial fluid levels of microRNA 126.3p, and interestingly, contrary to what we expected, it is not increased in synovial fluid. So we have high levels of 126 in the blood, but not in the synovial fluid of individuals with established osteoarthritis. And so we're not sure why that is. But what it tells me is that microRNAs are very tightly regulated. And the levels that you find in a particular biofluid or tissue, in my opinion, is perfectly designed the way it's supposed to be. So it's not by chance, it's tightly regulated. so that whatever function it's meant to accomplish is being accomplished. So I think different biofluids will tell you different stories about microRNAs, and this just brings us back to the complexity of osteoarthritis. Depending on what biofluid you want to measure, you may get a different answer for your microRNAs. And so the approach we're taking in my lab is to be very thorough. We are looking at as many biofluids and as many tissues as we can to get the most comprehensive and whole picture we can about the role of 126.
Wow. Thank you for explaining all of this. I really appreciate it. We have a question, again, from the audience. If we want to target MR126-3P, then should antagonists be delivered systemic through blood or intertracheal? Sorry.
That's a great question, and I think I can understand what the audience is saying.
Okay.
So this is a really sophisticated audience, and I'm really impressed with these questions, because that's exactly the question that we had and that we are currently aiming to answer with our animal model. So I mentioned that we have mice that we are currently inducing osteoarthritis, knee osteoarthritis, and we're actually investigating two different methods of delivery, systemic and intra-articular. So what that means for systemic delivery, we are administering the mimic, or the microRNA, or the inhibitor, or an antagonist here, as asked in the question. We are administering that into the blood. So that would be systemic delivery. And we are asking the question, does the knee osteoarthritis get better or worse with each one of those treatments? But we're also using an intra-articular delivery method. Now, if you can imagine, the mouse knee is super, super small. So we have a very talented postdoc in my lab. Her name is Dr. Madhu Bagel, and what she's doing is taking a very small needle and injecting a very small amount right into the knee space. What that's going to allow us to do is determine if we can actually modulate the levels within the knee joint to improve knee osteoarthritis and not have to administer the drug throughout the entire body. As you can all imagine, the more local we can provide the treatment, the more we can target it to the knee joint, the less chances there are that it's going to have other negative effects throughout the body. And so we're interested in looking at both of these delivery systems to ask the question, how can we better target knee osteoarthritis? How can we get the best possible outcomes?
Okay. We have another audience question. Have you found this elevated microRNA to be associated with any other specific inflammatory conditions?
That's a really important question, because that's the thing about microRNAs. They are associated with many things. So as I mentioned, there's already a lot of previous studies that have looked at microRNA 126 in the vasculature. So it stands to reason that there could be some association with this microRNA with other types of conditions. One of the things we're really fortunate to be able to do at the Henry Ford Health System is have really rich and comprehensive clinical data from our patients. So when we recruit or invite someone to participate to our study, what we're asking is for a really comprehensive medical history. So that allows us to know, do these individuals have other comorbidities? Do they have other conditions? So we've been able to analyze our microRNA 126 profiles in the blood, not only against knee osteoarthritis, but against a variety of different conditions. And we have yet to find any association that's as strong or as specific as we have found for microRNA 126-3P and knee osteoarthritis. That doesn't mean that it's not associated with anything else. It means that for the handful of things that we've looked at, we're not able to find any other associations. And I should also note that the things we're looking at are the comorbidities that are really prevalent in the knee osteoarthritis population. So things that are associated with metabolic disorder, for example, diabetes. And so we're not finding any association of microRNA 126-3P with those types of diseases. So we think that it might be specific to knee osteoarthritis. The other thing I should note is that we've also looked at other factors like age or sex or BMI. And these are all factors that are known to impact microRNA profiles, generally speaking. So it's important that we consider those factors when we're looking at our population. And so far, we can't find that those are the explanation for the elevation of microRNA 126-3P that we've observed. And again, this is really encouraging us and suggesting to us that this microRNA could be specific to knee osteoarthritis.
Okay. Thank you so much for all that information. Thank you for the audience for submitting those questions. We're going to move on to a Q&A session. I'm going to throw out the first question to both of you. What challenges are researchers currently facing in advancing our understanding of this disease?
I'll start with that briefly. I think one of the things that's a major challenge in the field is grouping people with osteoarthritis into a single category. As I said, and as Amanda said, there's many reasons why someone would get osteoarthritis, and I think it's very important to separate, to try to understand why those people have osteoarthritis and then separate them into different study groups. And I think that's going to make all of our studies a lot more powerful when trying to understand either the genetics or the biology of why someone develops OA. So I think this idea of subgrouping people based on the type of OA that they have is a challenge. It's hard to do, but I think it's one that people are working on, and I think will greatly improve a lot of the work that we do.
I would have to echo that. I think the biggest challenge is understanding the different subgroups and the types of people who have osteoarthritis. And all of the ones we've talked about, Mick talked about genetics and heritability of osteoarthritis. The mouse models we're looking at are traumatic. So this means that we use surgery to induce osteoarthritis. Those are just two types. I briefly mentioned metabolic disorder. That's another entire group or category that we know increases your chances of developing osteoarthritis. And so I think understanding the etiology or the cause is part of it. And then the progression is another part of it. Why do some people progress faster than others? And so I think all of this is to say it's a really complex disease that not only has multiple different factors that could be causing it, but I think there are also factors that impact the way it progresses. So that makes it seem that there will also be different factors that impact the treatment. I suspect there may not be one magic bullet treatment for everyone to improve their osteoarthritis. I suspect there may be a bunch of strategies that we've shown that are effective, and that it's going to take some kind of like holistic approach really to managing this disease, and hopefully one day finding a cure.
Okay, perfect. How can we as patients contribute to research efforts?
I'll start with that again. I think trying to find studies and then getting involved with them. I need to say thank you to all of those of you out there who have participated in studies. It's tremendously valuable to us to have you participate, right? So asking us to sequence your DNA, collect blood, collect urine is the basis of our studies, right? And we couldn't do the work that we do without you. And so I think we might need to be better in trying to advertise these opportunities to you, but I think reach out to your provider. If when you go see your doc next time, and just ask him, are there any studies that I can be involved in? I have arthritis. They're likely to know. I collaborate with a number of different surgeons in my department, and we have several different studies ongoing. And so, yes, I think basically reach out to your providers, and realize that it's very valuable to us that you do participate, and we do thank you.
And I'm just going to echo that again. So the Henry Ford Health System, we're really fortunate to have people who are willing to participate in our study. All of the findings that I presented today related to microRNA 126.3p would not have been possible without individuals volunteering to participate in these studies. And so currently, when someone comes into the hospital and they're having either a joint replacement or having arthroscopic procedure done, we consent them and ask if they're interested in participating. And more often than not, people say yes, and we're very grateful for that. And so involvement in research is really important. And the other piece I would add to that is really advocating for yourself and having your voice heard. And so I really try to engage with the patient population that we're trying to help, because we want to understand what are the symptoms that are bothering you, what is your experience, and how can we address that? Because at the end of the day, even if your x-ray looks better, that's not going to matter if you're having a lot of pain and a lot of functional limitations. What matters is your quality of life. And so we always want to remember that that's what we're really trying to work towards. And without patients really advocating and telling us what their experience is, I think that becomes so much harder. And we may lose sight of the real goal, which is to help people.
Okay, so we had an audience question come up. Where do you see the future of OA management heading towards? Is it personalized medicine, genetic intervention, or natural remedies that might be able to influence microRNA?
Well, I'm going to assume that microRNA piece means the question's for me. That's a great question. And I'm biased, so I think microRNAs are the way. No, I'm just joking. There's going to be a combination approach, right? So I don't think it's going to be one thing necessarily, but I am a huge fan of personalized or precision medicine. This ties back to what Mike was mentioning about there being so many different types of individuals with osteoarthritis that I think we really need to treat the patient and not necessarily the population as a whole. So one of the things that really is fascinating to me about microRNAs is can we use the same marker to identify a person, let's say by measuring the levels of microRNA in their blood, and then can we understand what that microRNA is doing? Let's say it's impacting angiogenesis or some other mechanisms that we know can be contributing to the disease. And then can we now target that specific microRNA to correct that process and hopefully improve outcomes in disease? So the reason that's interesting to me is because now, instead of just administering the same treatment to everyone, what we can do is administer the treatment to the individuals who have that specific cause of the problem. So hopefully that makes sense in tying those three pieces together, which I didn't really mention in my presentation because we're not quite there yet, but that's the goal. That's the overall, in the grand scheme of things, that's the goal that we're working towards, is really how do we treat individual patients based on their profiles. And so I am a fan of precision medicine, and I do hope that that's the direction we're going in. But that being said, I think there are many different interventions that are currently being explored, all of which are equally important because we may need a combination approach.
That's a great answer. I know I have rheumatoid arthritis in addition to my osteoarthritis, and they obviously have biomarkers for that, so I'm able to be on medication for it. But I think that
Exactly.
doctors are just frustrated when they go, we don't know what starts this, we can't stop it, and good luck. I did have a question about, like, I know people who are, so I'm 40, and I know people who are a little bit younger than me have been told you're too young for a knee replacement, shoulder replacement, some sort of a replacement. Would you follow that? Like, if your research is where it is right now, do you think eventually there are some doctors who would say, okay, so there's research out that's saying that you have this, it doesn't matter what your age is? Because some doctors here, in the US, I'm from New York, and it's a very orthopedic century state, unfortunately. So a lot of osteoarthritis, but I've had people who suffered more than me at a younger age, and they were told by their doctors, you're too young for a knee replacement. And that was the end of the conversation.
Yeah, that's really interesting. I can't quite comment from a surgeon perspective, as again, I'm not a medical doctor, but it's difficult because we actually, when we find families, some of them are like your family, like my family, where you're experiencing symptoms in your late 20s, right? And that's quite early for in developing OA, and then eventually you have to have surgery. I had a fusion of my toe joint in my mid 30s. So yeah, I guess my advice would be maybe get a second opinion, because we all know that this disease is very painful, right? And yeah, maybe reach out to another provider and see, get their opinion, what it might be. These joint replacements don't last forever, right? So if you are young, you get a joint replacement, it's likely you're going to have to get a second replacement. The technology isn't quite there where it's going to last you an entire lifetime. So that's another thing to consider as well, and maybe what some of these surgeons are taking into account.
Okay. And I don't know if either one of you have an opinion on this, but do you have thoughts regarding stem cell therapies?
I can offer some guarded thoughts, some, I guess, very general thoughts. I'm not sure that there's enough research to support the use of stem cell therapies for osteoarthritis. So I'd be very cautious if you see a kind of claim that there's a stem cell therapy that can help treat this disease at this time. There is a lot of fantastic research that's being done. It's in the pipeline, and so we do hear updates periodically on, even first in human stem cell therapies for knee osteoarthritis. But at this time, I'm not aware of any approved therapies for stem cells. Is that fair, Mick? Anything else?
I would completely agree. I think, as you said, it's being worked on, and I think there's potential, but as of right now, yes, the data just isn't there.
I'm just gonna thank you everybody so much for coming. I'm just gonna pass it over to Laura, and she's gonna wrap up things in a minute or so.
Yeah, absolutely. I want to thank everyone first and foremost for all of the great questions that came in. I'm sorry we weren't able to get to all of them, but again, please feel free to email us at info@curearthritis.org, and we will get as many answers back to you as we can. I also want to say thank you everyone who submitted questions, and absolutely to our fantastic moderator, Tracy. Wonderful job today, Tracy. We so appreciate you being here and sharing your story. Also to our fantastic researchers for sharing their innovative work towards osteoarthritis. We are eternally grateful for you. You can see firsthand the impact that ANRF has in supporting these incredible individuals on their quest for a cure. I would once again like to thank our partners, Amgen, AbbVie, AstraZeneca, and AOPI for your support of this webinar. And lastly, thank you to everyone who joined us for this great event. If you enjoyed this webinar, we kindly ask you to consider making a donation today by visiting curearthritis.org/donate. Every contribution helps. We will be sending live attendees a survey after the event. Your feedback helps us find a direction for future webinars. Please share your comments with us so we can continue to provide important and relevant content to you. And please don't forget to sign up for our other Researcher Spotlight webinars. Thank you all for coming, and please remember to follow us on social media to stay well connected. Have a nice day.